We're talking about chapter 20, which is another good one because I get to clear up a lot of misconceptions. Talking about antibiotics, or antimicrobial drugs. Alright, so the broad term for a drug is a chemotherapeutic agent. And this is anything that you could possibly use to treat things that are wrong in the human body. And there's lots of different chemotherapeutic agents out there because there's all sorts of different things that can go wrong with the human body that you can treat with medicine. What we want to focus on in this chapter is obviously antimicrobial drugs. Right? Those drugs that are used to treat or kill pathogenic microorganisms. And one of the major misconceptions we'll tackle is the fact that the vast majority of antibiotics only work against bacteria. And we'll see why. Okay, as for the antivirals, antifungals, and antiparasitics, those are going to be our loose ends, because they're not as common. Alright, so next misconception. Okay. Where do you find antibiotics? Okay, people don't often realize that most antibiotics are naturally derived. We find them in the soil, okay, from soil-producing microorganisms. Once we find new natural antibiotics, it's become commonplace to chemically modify them. Okay, semi-synthetic drugs are very popular because when you chemically tweak antibiotics, you can improve their effectiveness, which we'll see an example of later on. And then the one that seems like it's obvious is the rarest of all. Synthetic antibiotics are not common. Okay, very few effective antibiotics are man-made. Most of them are, again, naturally derived. But these are drugs we typically find from soil microbes that do more damage to pathogenic bacteria than they do to us. Okay, so another thing to keep in mind with this chapter. I cannot stress enough that antibiotics are anti-life drugs. Okay, yes, ideally they do more damage to the pathogen, but they also do some damage to you as well. Alright, so quick backstory. I won't spend much time on this because we're already behind. Talking about the brief history of antibiotics. Right? What were the first who discovered them? Alright, so Paul Ehrlich, was it 1910, reported the first antimicrobial agent, salvarsan. Okay? He was trying to treat syphilis. And he was trying to come up with a magic bullet, okay, chemical that would work against all possible pathogenic organisms. And he knew that arsenic was very effective at killing things. So he utilized an arsenic-based powder called salvarsan. Fairly toxic, but was somewhat effective against syphilis. Alright, about 20 years passed, we got Gerard Domjak. He came up with the first antimicrobial drug, Prontosil. Now, the funny thing about Prontosil is that in vitro, so in a test tube, it was garbage. It wasn't effective at all. But when administered to a living organism, say in vivo, it was highly effective against streptococcal infections. So that's weird. Okay, right? It didn't work in a petri plate, but it worked great in a human being. Turns out Prontosil was not the drug. It was, in fact, sulfanilamide or sulfonamide, sulfa drugs. Okay? When the people were ingesting Prontosil, their bodies were metabolizing it, turning it into the antibiotic, sulfonamide. Okay? 1932, still used today. Okay, this is a highly effective inhibitor of folic acid, but we'll come back to this guy later on. So that was the first antimicrobial drug reported. Alright, around the same time, we had Alexander Fleming. Okay, he discovered, as most people know, penicillin. And as a lot of you have heard the story, it was purely accidentally because he had a contaminated petri plate. Okay, he had inoculated his plate with bacteria, but it became contaminated with a fungus. And the funny thing is, around the fungus, there was this clearing. zone where the bacteria were killed off. So he's like, what the hell's going on, right? What is the fungus doing to kill the bacteria? Well, when he took a deeper look, he found out the fungus was producing a natural antimicrobial compound, penicillin. Okay, so this was produced by the fungus Aspergillus penicillium. So Fleming gets all this notoriety for his accidental discovery of penicillin. Here's the funny thing, though. The next two guys probably deserve a bigger pat on the back, and you've never heard of them. Ernst Chain and Howard Florey. Okay, this is the 1940s, World War II. They figured out how to mass produce penicillin. Right? That's a big thing, figuring out how to mass produce antibiotics. So they laid the foundation for that. Really helped the Allies in World War II. But their methods were so effective that penicillin was sold over the counter. Within 5 years, we saw the first cases of antibiotic resistance to penicillin by bacteria. You'd think we'd be a little bit more cautious when we saw that back then, but not really. All right, so again, taken from this historical fact, we know that microorganisms can produce natural antibiotics. So again, we go looking into the soil. So why the soil? Most antibiotic-producing microorganisms are found in the soil because the soil has, gram for gram, the most bacteria in it. Okay, the most microorganisms. And if you've ever had an ecology lecture, and they talk about what happens when you put a whole bunch of organisms together, you have to talk about competition. Right? Organisms fight for space. They fight for food. Bacteria are no different. Okay, they fight for real estate. And how do they fight? They produce antibiotics. So, examples would be bacteria like Streptomyces, have given us some of our most potent antibiotics. Or our friends, the Bacillus species, like Bacillus subtilis or Bacillus cereus, have given us a few as well. But it's not just bacteria. A number of fungi produce antibiotics as well, like the penicillium and the cephalosporium. So this is just a reference to kind of relate a number of common antibiotics to the organisms we discovered them in. So right at the top, bacitracin. If you've ever used Neosporin or triple antibiotic ointment, you have used Neosporin. Or sorry, you've used bacitracin. Bacitracin comes from our friend B-Sub, same one we use in lab. Some other big ones, chloramphenicol comes from a Streptomyces venezuelae. Guess where that was discovered. And then fungi at the bottom. Okay, so the Streptomyces are really weird bacteria, but they have given us some of our best antibiotics. So there's a bit of a gold rush right now. Okay. There has not been a new novel antibiotic discovered in 40 years. We're getting desperate. Okay, because if you take a look at the chart here, long story short, you see the bars are declining over time from the 1980s up till almost present. That's showing you the number of new antibiotics reported. Okay, and if you looked at today, the bar would be even slimmer. So there's a lot of research right now going out there, digging in the dirt, trying to find newer, better antibiotics. All right, so the next thing we're going to look at here briefly is looking at some of the features of antibiotics. What are some of the characteristics you look for? All right, so first things first. Antibiotics are anti-life chemicals. Okay, but ideally, they have selective toxicity. It's all in the name, selective toxicity. They're selectively toxic against bacteria, and to a lesser extent, you. And why are they selectively toxic? Because antibiotics target different features of bacterial growth. Okay, and you want to target something common to bacteria and lesser so to humans. Okay, so that you don't feel worse than you already do. Now, this selective toxicity is measurable because antibiotics vary greatly. We measure it as the so-called therapeutic index. So I have to read this because I never remember. To figure out the therapeutic index, you need to take the lowest dose toxic to the patient. Okay, so how much can you give somebody before it becomes poisonous? Divided by the dose typically used for treatment. So big number divided by little number. That gives you an inverse scale. Okay, so a high therapeutic index. These are ideal antibiotics. They're highly effective against the bacteria, and they do less damage to you. Okay, low chemotherapeutic index. These antibiotics tend to be drugs of last resort. Incredibly potent, but they can be quite damaging to you as well. Okay, so inverse scale. High, good. Low, less good. All right, antimicrobial action. Most folks think that antibiotics just outright kill bacteria. Most don't. Some may just merely inhibit growth. Okay, so are they bacteriostatic or bactericidal? Okay, many bacteria or many antibiotics are purely bacteriostatic. Okay, all they do is slow down the pathogen. Now, this is still great for you because it helps the immune system catch up. Now, some of the more potent antibiotics, and sometimes fairly toxic, are bactericidal. Okay, cidal implying they outright stop or kill the organism. Okay, so some are bacteriostatic, merely slowing growth, while others are cidal, where they outright kill the organism by stopping its growth. All right, spectrum of activity. People often think that antibiotics are magic bullets, right? They work against all possible bacterial pathogens, and it is so very far from the truth. So, considering spectrum of activity, okay, many antibiotics are purely narrow spectrum. Okay? And so, that's like saying they only work against gram-positives and not gram-negatives. Right? Penicillin is a great example. Penicillin is fabulous against gram-positives, but it really doesn't do much against gram-negatives. Okay, it's fairly narrow spectrum. Now, these drugs are ideal for a couple of reasons. One, they're typically used when you know what pathogen you're dealing with. Right? Do a rapid strep test, run a gram stain, whatever. Okay, you get a rough idea of what you're dealing with. Because one of the messages we want to really hammer home today, something I want you folks to consider, your gut is full of beneficial bacteria. You need them, they need you. When you take antibiotics, do you think your gut bacteria are spared? No. And that's something we're looking long and hard at right now in research, is how impacted are they when you take various antibiotics? And does that have any sort of long-term implications on your health? So on that note, looking at broad spectrum drugs, you don't know what pathogen you're dealing with, so you got to rely on an antibiotic that's going to hit them all. But it comes at a cost, right? It's going to more greatly diminish that microbiome in your gut. And that can lead to long-term issues, which we'll see here in a little bit.
Aren't the microorganisms in the human gut, wouldn't it be different from the pathogens that come from outside? So couldn't a drug be created that would only target certain chromosomes? The ones there are human chromosomes in the bacteria of the gut. So
Well, hang on to that thought. Okay, hang on to that thought. She's asking if we can make more fine-tuned antibiotics. All right, so looking at spectrum of activity. They're all over the place. That's what I'm trying to show you here. And, okay, we've seen gram-negative, we've seen gram-positive, we've seen mycobacteria, right, the acid-fast guys. I know chlamydias and rickettsias are separate. Technically, they're gram-positive, gram-negative. They reproduce in slightly unique ways, especially the chlamydias. You don't get into those until you take advanced micro. But, look at tetracycline there. Right? That bracket covers all but the mycobacteria. Very broad spectrum. You see the penicillins in the middle, really only good against gram-positives. And some get ridiculously narrow spectrum because, Take a look at isoniazid on the left there. The only time you'd ever take isoniazid is if you had tuberculosis or leprosy, because it's only good against mycobacteria. Okay, so they all have different spectrums of activity. All right, next thing to consider. What happens when you take multiple antibiotics at once? So combinations. Okay, you have to consider the interactions of these drugs together in the human body. So, in a hospital setting, it is not uncommon to give an individual 2 or 3 antibiotics at once in the appropriate situations. Ideally, when you use antibiotics together, they are synergistic. Okay? Synergism means they're improving upon each other. They're helping each other out. Right? That's ideal. That's obvious. Now, apparently, and I've yet to find any really good, decent examples, there can be situations of antagonism. You use the antibiotics together, they hinder each other in some way. But again, these examples are incredibly rare. And then you have additive, where they're used together and they don't really influence each other. It's also a possibility, but most of the time they're synergistic. But you do have to take care. All right, here's a good one. Tissue distribution, metabolism, and excretion. Okay, so what happens once that drug gets into your body? All right, so first things first. Site of infection must be considered when choosing your antibiotics. Okay, will the antibiotic get to where it needs to be? So, for instance, some antibiotics are not pH stable. They can't be taken orally. They'll be destroyed by the stomach. Okay, so they have to be administered intravenously. An even better example. Take a look at spinal meningitis. And you get inflammation and infection within your central nervous system. You have a big problem. The blood-brain barrier. Okay? Triple set of membranes must be passed. Most antibiotics cannot pass the blood-brain barrier. They're way too polar. So, you must choose then an antibiotic that's extremely non-polar. And that list is real small. Okay? Really, the only great example is rifampin, rifamycin. Okay? These guys can pass through the blood-brain barrier and get to the central nervous system. Rate of elimination. Right? How long before that antibiotic gets excreted, gets urinated out? So that is measured as half-life. Okay, and the half-life of anything is how long does it take for half of something to go away? So here is how long does it take for half of the original dose to be excreted? This is important to know when figuring out how often to give a dose and how high of a dosage to give an individual. Okay, and antibiotics are all over the place here. So penicillin V. Okay, this was the second penicillin discovered. You had to take it every 6 hours and a reasonably high dose. Okay, that's a lot of doses in a day. Azithromycin, on the other hand, is the so-called Z-Pack, which by now most of us have had at some point. And if you've had it, you know you take 2 on the first day, and then 1 each day after that, right? That's a 24-hour dose. That's pretty good. Don't have to remember to take it as often. Other examples get more extreme. I had a student years back, had terrible eye ulcers, horrible eye infection. She had this little ice chest she had to carry with her for a couple of weeks. She had to leave the room every 30 minutes to dose her eye with some antibiotics that I had never heard of. But the dosage was extreme. Incidentally, her eye made a full recovery, which was amazing. All right, so last thing to consider, adverse effects. So when you watch any commercial for any sort of prescribed drug, they talk about how fabulous it is, and then somebody rattles off a bunch of horrible side effects, right? This is no different, folks. These are prescribed drugs. So, for instance, toxic effects, side effects. Okay, many antibiotics can have some rare but scary side effects. So, streptomycin amongst a few others, can be ototoxic. Okay, they can damage your inner ear, so you can have impaired balance, deafness, and in the case of streptomycin, kidney damage. About 2 years ago, I was given a fluoroquinolone, and for me, it was ototoxic. I still remember I was standing here teaching, and my ears were screaming. I called, like, what the hell's going on? They're like, oh, just stop taking it. It should resolve. I had vertigo for 6 months, and the tinnitus is all but faded it. I can hear it ringing just a little bit. But it can happen. I sat my butt on that desk for a whole semester. Because every time I stood up, I was like, ugh. But anyways, chloramphenicol, it's the drug of last resort. This is one of the scarier side effects. Aplastic anemia. Your body temporarily, as I've read, loses the ability to make blood cells. My wife has an allergy to penicillin. One alternative she was offered I had not heard of. I wish I'd written down the name, but I looked it up and it had a rare side effect of hepatic liver failure. Okay, so she didn't end up taking it. I found out there's a new beta-lactam antibiotic that's actually safe for people who are allergic to penicillin. So she took that. No issues. It was pretty cool. And there's all sorts of others. Allergic reactions. You know, you put anything organic in your body, you could potentially be allergic to it. Everybody has different hypersensitivities. Okay? Now, people range in how hypersensitive they are. So my wife, she'll break out in hives if she takes penicillin. But other people can have full-blown anaphylactic shock. Okay? It all depends on the individual. All right, now, hammering home that point I was making before. Right? You take antibiotics, you will diminish your gut bacteria in some way. The problem is that with the overuse of many antibiotics, or if somebody is taking it for too long of a period of time, or at really high dosages, you can kill back your gut bacteria to the point where unwanted organisms move in. Okay, so antibiotic-associated colitis. Many of you have heard of C. diff. Okay, this is the organism Clostridium difficile. Okay, every human being has some C. diff in their colon. It's just kept in check at basal levels. The thing is that C. diff is multi-drug resistant. So if an individual's gut bacteria gets killed off too much, C. diff says, hey, look at all this free space, and takes over. Your body does not react well. Okay, it triggers chronic inflammation. The organism can do tissue destruction or cause tissue destruction. And you can see in the image in the bottom right there, okay, that's C. diff colitis. Okay, compare it to the left. Nice, smooth, clean tissue. Traditional therapies to treat this, because of its multi-drug resistance, have gone as far as segmentation of the colon, colostomy bags. Some newer treatments, though, have been somewhat successful. Believe it or not, there are prescription-strength probiotics, like VSB? I always mix it up. Where I looked it up, the CFUs, the number of cells per pill, number in the trillions. It's obscene. But that's had some success. I'll talk about fecal transplantation in the last week, where people are receiving quite literally poop samples from family members implanted into their colon, and that has wildly great success in actually repairing the gut microbiome, as gross as it sounds. Okay, so these are a lot of the characteristics to consider with antibiotics, right? So, selective toxicity. Is it bacteriostatic or bactericidal? Spectrum of activity. What happens when you use combinations? What sort of side effects will it have? And how long is it in your body? Now, the next part we're going to look at is usually pretty enjoyable, because folks often don't have any idea as to how antibiotics actually work. Right? How do they kill or slow bacterial growth? Well, superficially, all antibiotics target something about bacterial growth. Okay, so as we go through this, do me a favor. I want you to make it a challenge to yourself. Try to think of something I missed regarding growth, something essential for growth that's not covered. So try to see if I'm missing anything here. Okay, so we're going to target things that should be relatable because we've already talked about it throughout the lecture. Okay, so we've got 5 classes of antibiotics. Each class targets a different aspect of growth. So the first class, and the most common example, are antibiotics that target cell wall synthesis, meaning antibiotics that target peptidoglycan. Right? Peptidoglycan is very important to bacteria. It makes up their cell wall. It is their armor at the microscopic level. If you exploit that peptidoglycan, you will kill the cell. So these drugs often have a high therapeutic index because here we're targeting peptidoglycan, something you don't find in humans. Okay, something only common to bacteria. Now, if this was like a full-fledged pharmaceuticals class, I'd have to go through all the major families in this group and talk about them one by one. I don't have time for that. So with each class here, I just want to look at a few examples. Just to show you that while they are in the same class, they work in different ways. Okay, so let's start by drawing up peptidoglycan again. I know it's been a month and a half or so. Remember, we got all these layers. In between the layers, we had that glue. These tetrapeptide bridges. All right, so first example. It's very relatable. The penicillins, also known as beta-lactam antibiotics, which we'll talk about more in a second here. Penicillin does one fairly simple thing. It interferes with the enzyme that makes those tetrapeptide bridges. So penicillin takes away the glue between the layers. Right, well, big deal, right? You still got all these glycan chain layers. Well, take a look at the pictures. Bottom right or left-hand side, man, we've got bacilli there. Happy, healthy, don't know what it is. But look at the image next to it, where the cell has been ruptured. Okay, those have been treated with penicillin. You can see the cell wall has been weakened to the point where they explode. Vanco. Vanco is a very potent antibiotic, and Vanco works just by preventing the NAG and NAM bonds from forming. Right? With each glycan chain, it's NAG, NAM, NAG, NAM, NAG, NAM. They just can't be bound to each other. So no glycan chains, no cell wall. And then we got bacitracin, right? Topical cream, found in Neosporin. Bacitracin interferes with the membrane. So it's hard to see here, but we got those NAG and NAM subunits. They need to get out of the cell. Bacitracin prevents the NAG and NAM subunits from leaving the cell, so you can't make a cell wall. Penicillin, vancomycin, bacitracin, they're all in the same class. They all interfere with cell wall synthesis, but in different ways. All right, so like I said, in pharmaceuticals class, you go through each of the major families one by one, talk about what they do and how they're different. I just want to briefly do that with one group, the beta-lactams, the so-called penicillins. So what does beta-lactam mean? Well, you have to draw the structure of the average beta-lactam. So you got a house with a garage next to it and then a side chain. Right? This is the core nucleus of most beta-lactams. The square part here is called a beta-lactam ring. That's where the name comes from. Technically, it's like a little nitrogen there, but I don't get into the details. But that's the only thing that they all have in common. Okay, they got this square ring. All right, so there's a lot of examples here. They all do the same thing. Right? They all prevent the glue from being made between the layers. They all interfere with tetrapeptide bridge formation. And the funny thing is, the name of the enzyme that makes these bridges, the penicillin binding protein. Because we discovered penicillin before we discovered the enzyme that makes the bridges. So penicillin binding protein is what gets targeted. All right, so looking at the chemical structures on the right here. A little bit of story here. So we've got penicillin G and penicillin V. Penicillin G is the original penicillin. Okay? It had one big drawback. It had to be administered intravenously. If you took it orally, the stomach acid would destroy it. But then penicillin V showed up and changed everything. Penicillin V could survive passage through the stomach. And the chemists were like, all right, what's the difference? Why is this pH stable? And what did they find? While the common nucleus was the same, the highlighted part is the same, penicillin V had one extra oxygen in that side chain. And they're like, well, hey, there's a tiny bit of chemical difference here. It made it way better. What if we decided to continue chemically modifying it? Maybe we can improve upon it further, and that's exactly what we did. So below it, you have two examples of semi-synthetic beta-lactams, oxacillin and ampicillin. And as you can see, the side chains are vastly different. So in oxacillin, its side chain makes it resistant to beta-lactamase, penicillinase, as we're working with in lab. And with ampicillin, its side chain has made it extended spectrum. It's not just effective against gram-positives, it's also effective against gram-negatives to a certain extent. Okay, and that's something I forgot to mention. These drugs tend to be more effective against gram-positives than gram-negatives. And the story goes on. So, it's an old diagram, but I like it because it illustrates kind of the history of the beta-lactams. Going from first generation to now fifth generation. And you don't have to know all these. I just like to show you that how the little changes accumulate over time until you get to ceftaroline, that fifth generation that I had to add. So that mess in the upper right-hand corner is still a beta-lactam because it still has that ring. But everything else about it has been totally modified. So why did I take the time to add it? Because of MRSA. MRSA is a great, relatable pathogen in this chapter. Because MRSA is methicillin-resistant Staph aureus. Okay, normal Staph aureus, like I've said in the past, if you say the word penicillin at it, it dies. But MRSA laughs in the face of penicillin. Calling it methicillin-resistant isn't fair. They should call it beta-lactam-resistant. Because guess what? Pick any of these, except for ceftaroline, they don't work against MRSA. Not a one. So ceftaroline is one of just a few fifth generation beta-lactams that were fast-tracked by the FDA and whatever the European version is because it was effective against MRSA. I believe there's only 3 out there. So, highly guarded and protected antibiotics. Alright, so I know I spent a lot of time with that first class, but that's the most common and relatable one. The second class is also fairly common. We have a lot of examples here. So in the second class, these are antibiotics that inhibit protein synthesis. So put it another way, antibiotics that target ribosomes. Now, this is a great group, and there's lots of examples because compare ribosomes of humans to bacteria, right? In bacteria, it's 70S. In humans, it's 80S. The ribosomes are different enough that these antibiotics can tell them apart, to a certain extent. Now, there's tons of examples here, but usually, if you want to keep it simple, you break them down into either 30S or 50S inhibitors, because they bind to different parts of the ribosome. All right, so a couple examples of 30S inhibitors. Tetracycline and streptomycin. All right, so tetracycline here binds to the smaller part of the ribosome, and tetracycline prevents tRNAs from binding to the codon. Right? If tRNAs can't bind, that means no protein. Streptomycins are kind of clever. Streptomycins prevent the ribosome from finding the start codon. Right? If you can't find the start codon, it doesn't know how to translate. So no protein. Now as for 50S inhibitors, we've got chloramphenicol, which is a scary one. Chloramphenicol prevents peptide bond formation. So if the ribosome can't link together amino acids, no protein. And then I've got a fourth example here that's not listed in the picture. There's a good reason. Azithromycin. Right? The Z-Pak, which most of us have probably had at some point. Widely prescribed. We know it's a 50S inhibitor, but here's the funny thing. We still don't know exactly how it works. I check every year. Mechanism of action unknown. But we know it's pretty effective. All right, so those are the two most common classes. Those that inhibit peptidoglycan, those that inhibit ribosomes. The rest of them are few and far between. So the third class, antibiotics that inhibit nucleic acid synthesis. So we've got two options here. We can inhibit DNA, or we can inhibit RNA. All right, so inhibiting DNA synthesis, right? How do you prevent a chromosome from being copied? The only really good example, fluoroquinolones. Fluoroquinolones like Cipro, which is the big one, ciprofloxacin or moxifloxacin, they inhibit an enzyme called DNA gyrase. I hit you folks with enough enzymes back in chapter 8. Okay, I didn't want to add this one to the pile. But before replication, chromosomes are tightly wadded up, right? They're supercoiled. To be copied, the chromosomes have to be relaxed into that hoop shape we're familiar with. What does that? DNA gyrase. Okay, the human equivalent, if you've had 115, is topoisomerase. Okay, enzymes that just relax DNA. Fluoroquinolones inhibit this enzyme. So if you can't relax the chromosome, you can't copy it. And the cell dies. The next example is kind of a scary one. Rifamycins, or rifampin. These guys inhibit transcription. Okay, so they blind RNA polymerase. The only time you're taking rifampin is if you have meningitis or tuberculosis, because of all the nasty side effects you can get from it. But this is a very, very non-polar antibiotic. Okay, so this is the one that can cross the blood-brain barrier or penetrate into the very waxy mycobacterium. But typically folks will experience red man syndrome. This antibiotic causes everything about your body to become reddish-orange. Saliva, tears, fluid, your skin becomes flush, and you feel worse than you already do. Okay, so here we have two options. Target DNA replication or target transcription. Now the fourth class seems like it should be the obvious one. Inhibition of metabolic pathways. Right, well hell, shut down glycolysis, shut down Krebs. right? That'll kill them. Here's the problem. The funny thing about metabolic pathways from an evolutionary standpoint, they're pretty highly conserved. Glycolysis is glycolysis is glycolysis. Krebs is Krebs is Krebs. Compare humans to bacteria, it's basically the same. So here the options are limited. You need to find a drug that's going to inhibit a pathway common to bacteria and not to humans, but is essential for growth. So very few options. And the only good one I have for you is folic acid inhibitors. Okay, so why folic acid? Every organism needs folic acid to make nucleotides. The thing is that bacteria make their own folic acid, whereas we eat it. Okay, check the side of your cereal box, granola bar box, whatever. I guarantee folic acid is listed. You don't make it, you eat it. Okay, but bacteria don't have that luxury. They have to make it on their own. So, if you can prevent them from making it, they will die. So, we have two examples here. One we have already talked about in a previous chapter. Sulfa drugs, sulfonamide. And this time we're going to throw in trimethoprim as well. These guys are great examples of competitive inhibition of enzymes. And two, this is a fabulous example of synergistic antibiotics. Because sulfonamide and trimethoprim are still used today as a cocktail in Bactrim. Alright, so this is why they're used together. This is the pathway for folic acid. Okay, you have PABA, glutamate, dihydrofolate, folic acid. Okay, so it's a three-step pathway. So the sulfa drug targets the first enzyme in the pathway. Okay, competitive inhibition. Why is it competitive inhibition? Well, here's the sulfa drug. Here is PABA. That's the normal substrate. They look almost the same. The enzyme can't tell them apart, but when it binds the sulfonamide here, the sulfa drug, it gets shut down. Right? So it says here, shuts down enzyme 1, you think, all right, end of the story, organism's dead. It's not the whole story. Here's the thing. There's always some leaky expression. There's always some enzymes that don't get totally shut down, so it just pushes forward, albeit slower. So that's why we throw trimethoprim into the mix as well. Trimethoprim binds to enzyme 3, shutting it down. So synergism. If you shut down two enzymes in the same pathway, it's going to be a lot more effective than just shutting down one, right? All right, so that's the only good example I have here. Folic acid inhibitors. And then the last one, the last class are antibiotics that interfere with cell membrane integrity. Okay, and you think, all right, this is another great example, right? You know, bind to the membrane, make it leaky, kill the cell. Here's the problem. Compare the membranes of bacteria to the membranes of humans. They're all phospholipid bilayers. Our membranes aren't all that different from that of bacteria, so these drugs would target both. So not ideal. And typically, these are used against gram-negative bacteria. Okay, like polymyxin B, one of the 3 antibiotics in Neosporin. Polymyxin B binds to the outer membrane of gram-negatives. Okay, doesn't do much to gram-positives, really just bounces off their peptidoglycan. So, you put polymyxin B on the surface of your skin, binds to these gram-negative outer membranes, the cells get leaky, and they die. So, it binds to membranes. It's safe for use topically because the cells at the surface of your skin are basically dead. It's not going to do much against it. Okay, but you would never ingest one of these drugs in this class because it'll just wreak havoc to your body. Bind to whatever membranes it wants, killing those cells as well. Alright, so 5 classes, right? Those that inhibit peptidoglycan, those that inhibit ribosomes, those that inhibit nucleic acid synthesis, those that interfere with metabolic pathways, and those that interfere with membrane integrity. Any questions about any of those 5 classes? Okay. So, the thing I wanted you to think about, have I missed anything regarding growth? Right? We got transcription, translation. We got DNA replication. We got the cell wall. We've got the membranes. What else gets copied in the cell? There's really nothing left to target, right? No major factors regarding growth. Nucleases? Oh, the nucleus? They don't have a nucleus. This is a bacteria. So, with DNA gyrase, though, we've covered the chromosome being copied. So, in the last 40 years, there has not been one new example of a 6th class of antibiotic. There's been a couple the last couple of years that are debatable, but now we're trying to make more customized antibiotics that target very specific enzymes. Like, I've seen a couple that target ATP synthase, because ATP synthase can be kind of species unique, but the problem is it gets really damn expensive making drugs that customized. So we're getting a little desperate. All right, now the one loose end deals with mycobacteria. Okay, thinking tuberculosis and leprosy. And these guys get their own unique set of antibiotics because of how unique they are. All right, when we talked about mycobacterium in the past, we were doing acid-fast staining. And we said this is the only group of bacteria that have a waxy layer surrounding them. So that waxy layer makes them incredibly difficult to deal with. For one, it's very thick and dense and very nonpolar. Okay, so most antibiotics can't penetrate, but 2, because of that waxy layer, they grow super slow. They grow so slowly that they just wait out the antibiotics. Right? So it's very difficult to treat things like tuberculosis and leprosy because of that. So a cocktail of first-line drugs are administered to people with tuberculosis. And this cocktail includes things like rifampin, that's the transcription inhibitor, streptomycin, that's an anti-ribosomal, and then 3 that only work against mycobacteria. We got isoniazid and ethambutol. Okay, these 2 interfere with that waxy layer. And then pyrazinamide's a funny one. Until about 1.5 years ago, we did not know how it worked. We just knew it worked. And I finally checked again, and I was like, oh, look, mechanism of action. They finally figured out it causes the inside of the cell to become very acidic, killing it by, I imagine, denaturing its proteins. But if you have tuberculosis, you're taking these 5. And you're not taking it for 2 weeks and feeling better. You're taking them for months, if not years. And if these don't work, there's a second line of even more potent drugs. All right, so I want to do the next couple slides real quick, and then I will be quiet. Let's see. All right. So, there's only a couple parts left of this chapter, okay? And I like to get through this part if possible, because in nature, thanks to evolution, wherever there's an offense, there's a defense. So if there's bacteria that produce antibiotics to kill other bacteria, you can guarantee that there's going to be those bacteria that can defend themselves against the antibiotics. So how do they fight back? Right? Where does resistance come from? Well, overall, you can have either innate, passive resistance, or you can have acquired or active resistance. Okay, so passive resistance is just the cell happens to be resistant for whatever reason. It doesn't actively react to the antibiotic. There's just something about the cell that makes it resistant. So gram-negatives. Gram-negatives have an outer membrane. Because of the outer membrane, that makes them passively resistant to antibiotics. Right? It's a second selectively permeable barrier. Now, mean for it to be that way, it's just they're lucky. Some organisms, like tuberculosis or Shigella, they hide within your cells. evading antibiotics. Okay, that's passive resistance. Now, active resistance is where stuff gets scary. And we say acquired because with active resistance, these are traits. Okay, traits found in genes that allow the organism to actively react to an antibiotic on purpose. And they're acquired because they can get passed around. Right? Conjugation, transduction, transformation, take your pick. Cells can teach each other these things. Alright, so let's go ahead and do this. We'll get out of the way. This is part of the recording. Alright, so what are the rather clever ways that bacteria can fight back actively against antibiotics? Well, the first way we've already talked about in lab. Make enzymes that destroy the antibiotics. Right, on pGlow, we got that BLA gene. BLA encodes for beta-lactamase. So this is what beta-lactamase does. It gets secreted. It targets the beta-lactam like penicillin. And what it does is it finds that beta-lactam ring that they highlight and snip. Just cuts one little covalent bond. Big deal, right? One covalent bond is gone. That destroys the antibiotic. It's rendered ineffective. So this is one example. Okay, there are other enzymes that can be secreted to target and destroy antibiotics. But this is active resistance, right? Making an enzyme that actively targets antibiotics. 2 is probably the most common example. Altering whatever molecule would have been targeted by the antibiotic. Okay, so sometimes the bacteria can change the shape of the protein, so it can't be bound. So with ribosomes, right, plenty of antibiotics can target ribosomes, but some bacteria can change the shape of the ribosome so the antibiotic can't bind. Or better yet, what about MRSA? Right, that whole, or all the penicillins, all the beta-lactams target the penicillin binding protein. But why is MRSA special? Why is it spared from having its penicillin binding protein inhibited? Because it makes a different version. That's it. It's the same enzyme, just a different version. And different enough that all the beta-lactams, except for that 1/5 generation, don't work against it. Third example, prevent the antibiotic from getting into the cell. Gram-negatives can shut out antibiotics by turning off their transport proteins. So for instance, they have these porins, these kind of universal transport gates. They just shut them down. The antibiotic can't get through that membrane. It's not going to hurt the cell. So this allows them to just wait out the antibiotic. The fourth example is the scariest. This is where we're running into the biggest problems with growing resistance. Rapid expulsion of antibiotics, so-called efflux. So efflux pumps we talked about with active transport. Okay, these are transport proteins that behave like sump pumps. Right? In the basement of a home, sump pumps pump out unwanted water. Efflux pumps pump out unwanted antibiotics. So as the antibiotic gets into the cell, popped right back out. Right? This is, again, particularly scary form of resistance. And the fifth example deals with the sulfa drugs here. Right? How do you get around a critical metabolic pathway that's been shut down? Well, for one, some organisms have detour pathways built in. Like, okay, fine, my central pathway shut down, I can go around it. How clever is that? Or they just say, screw it, I can't make folic acid anymore, I'm going to become an auxotroph and just get it from my environment. They can do that as well. Alright, so this is from a different book, but it really nicely illustrates the two sides of this coin. Right, on the left, we have all the different mechanisms of how we can inhibit growth. On the right, all the different ways that the bacteria can fight back against the antibiotics. And I get into this a bit more with the recording.